Alrighty, so now we're into the second segment of this lecture on species, what they are and how they impact conservation biology and population ecology and things like that. We ended with sort of an idea of lots of things like hybridization events and our understanding of the DNA evolution in regions impacting our definition of species beyond other clear situations like, does that thing look like it's a different species or not? We don't know. Gene flow helps us understand who is mating with who, biological species concepts aside, etc. Today, I want to dig into the evolution ideas a little bit more, and then we'll end with some microevolution, where we can see species shifting based on environmental conditions shifting. And that's you can really document that. Macroevolution is a little bit tougher. So let's get into that. There are three main ingredients of evolution now we're using this term natural selection or phrase. This is from Wallace. Remember, he was the one that submitted his paper, and he said, I think I know why all my birds look different or not down in the Pacific, South Pacific. It's because there's something called natural selection. They get to a spot and their genetics start, well, you didn't have the word genetics or even the concept. Mendel hadn't arrived yet. But they are being altered by their environment, or at least they're faced by pressure by the environment to either, let's say if it's a bird, grow out a beak that allows them to catch insects, nice and thin, or go after nectar like a hummingbird, extremely thin and long, or be huge like a cardinal and crack big seeds, okay? Those are different selective pressures that altered, in some sense, the shape of the beak based on what the bird needed to survive. Okay, that's natural selection. Three main ingredients. I've just pretty much given you an idea about these, but you have to have variation among individuals. So some birds, let's say cardinals, have bigger beaks than others. You have inheritance of that variation. All right, so some birds with bigger beaks reproduce with young that have bigger beaks. Okay, but some of the young don't. Right? Even with the one that had the big beak, it's young, there'll be variation of the children in terms of how big their beaks are. So it's not a one-to-one relationship. That's sort of Lamarckian. And then differences in fitness among the individuals is related to that variation. So if a bird has a really big beak and there's only big seeds around, it's going to be a lot better at getting resources and reproducing, and therefore sharing that character of a big beak into the next generation. So here we see nicely that there is this interaction between genotype or phenotype and the environment. How does that genotype help the organism survive in the real world? And the more individuals that are produced, the better off for that gene milieu, that individual's genetic component, to make it into the next generation. All right, so those three things: variation, inheritance, differences in fitness. I'm sure you've heard about that, and I'm sure you heard about it again, but I thought you should hear about it in this class as well, even though it's called ecology. And I want to hone in on something called rapid speciation, because I think it's a great model for understanding evolution and this interaction between the environment and the genotype, phenotype, and understanding natural selection. And we're going to look at a couple of examples. We won't look at fruit flies, but that's a classic. That's a classic. There are a lot of researchers that are looking at fruit flies on the Hawaiian Islands. Turns out there's not just the one you look in genetics lab, Drosophila melanogaster, but a whole bunch of different ones, and they tend to hone in on different islands or even different drainages in those islands, and those are unique species. So we're going to look at little bigger organism, the finches, and then also the anole. No, I don't think I'll talk about the anole lizards on the Caribbean islands, but they've done it also. And I'm going to end with the silver swords of Hawaii, just because it's a topic that I love. Darwin's finches. This is out of your book. It shows you a fairly nice example of sort of a phylogenetic tree. And you can see everything from the small ground finch down to a warbler finch. What? A warbler finch? Look at the beaks. Look at those beaks of the warbler finch. Those go after insects. This one is a vegetarian finch, just eats buds, flower buds. The woodpecker finch, I'll show you an example of why that's called that in a little bit. That's not a woodpecker beak, but it is actually, it's a smart bird. It uses a tool. And then on up to the seed eaters, all right? Note that the original ground finch, or the original finch, was probably a ground finch, and it flew over from South America to the Galapagos Islands. That's where the Darwin's finches are. Now, the species developed both allopatric, under allopatric conditions and sympatric conditions. So allopatric is with geographic separation. So some of these species populations wound up on different islands, and there was no longer any gene flow, and they became a separate species. And now you bring them back together and they can't reproduce. And their, let's say, beak size, if we're focused on that character, has changed dramatically, all right? And so you have new species. It can happen sympatrically also on the same island. There's just too much finch on an island, and the finches begin to, just by random behavioral chance, start to go after different resources. And so then you go from a seed eater to an insect eater, or at least one that's starting to explore it with insects and then discovers, wow, there's all sorts of insects. I don't have to compete with that big-headed guy over there for seeds. I could just go with insects and sky's the limit. Okay, that can happen within the same geographic location. Right, let me look at my notes here. Let me just give you a quick overview of how you read a tree like this, these phylogenetic trees, just a couple of points that are important. So this is out of your book, figure 717, and it shows sort of a cladogram of things from lampreys up to mammals. And what I want you to notice is there's a line over here on the left called time. So in the past, there was a common ancestor that had characters that basically carry all the way up to mammals, all right? Even though it looked a lot more like a lamprey than, in fact it did, than a mammal, all right? So time, you're seeing time on this axis, and so as you move across the tree, you're going from ancestral states, ancestral states. So this is an ancestral organism. It has some version of a nerve cord that goes all the way down, all right? But it wasn't until cartilaginous fish developed that you start getting some serious stuff that's almost like bones. Then you get the bony fish. I guess that's a bass, could be a trout, I don't know. Then you get lungs for air exchange instead of gills. And you can see this moving up. So lampreys don't have jaws. Cartilaginous fish do. Bony fish, you get a bony skeleton. Everything after that bony skeleton event, that mutation that led to the bony skeleton, mutation and selection on the mutation, everything after that has bony skeletons. Everything after the development of lungs has lungs, okay? And some amphibians have gills at the natal state, but by the time they become a frog or a salamander or something like that, they've got well-developed, lungs. Then you have legs, the egg, milk, and hair. I love the fact that this particular image shows a goose here next to the reptiles. They weren't quite sure where to put that thing. Closer to reptiles, I guess, they just left out dinosaurs. Okay, so and then that last little segment, as you go up that the figure on the right side with those yellow highlights, you see what unique character states developed that probably led to the evolution of the whole group. downstream. So you go from no jaws to jaws, no skeleton to skeleton, no lungs to lungs, etc. Okay, and so you can map those character states as they evolve on a tree like this. The main thing for this class, though, is that there's a evolutionary history you're seeing from primitive states to derived states. We mammals are quite derived compared to fish, right? Okay. Now we have allopatry and sympatry. I did talk about that already. This is out of your book, figure 718 and figure 720. And here, allopatric speciation, I told you that happens when you've got a geographic separation. So you have these little mice on the landscape, a river develops, a river runs through it, excuse me, and they're separated. And when they used to have, they're using an example of coat color, two different coat colors all intermixed. After that river develops, they become separate. And if it's over a long enough period of time, if the river disappears and they come back in contact, they cannot share genes anymore because they no longer can breed. So that's allopatry. It was caused by a barrier, a geographic barrier, separating the two subpopulations, which then become two species. Sympatric is within the same location. So this is, let's say, Lake Victoria. It's a great place for evolutionary studies because of all these crazy cichlids. And they're all within the same lake, but they're all very different, and they've evolved different habits. I'll show you an example in a little bit that has to do with what they eat and what the mouth parts look like and how that might have happened. But this is sympatry, evolution within sympatry of new species. They don't interbreed. Some may interbreed more than others with hybrids because they've only recently evolved into separate units. But some of them did that a long time ago, and now they just can't interbreed. It's too late. So that's sympatry within the same geographic location. I'll come back to that in a little bit. Oh, maybe right now. So once again, allopatry with geographic separation, sympatry without, and allo means different, sim means the same, right? Can sympatric speciation occur in an allopatric setting or way? Yes. And so in my world of plant pathology, there's a fungus called Heterobasidium anosum. And there are strains that are called SP and F for spruce, pine, or fir. And you have strains of a Heterobasidium in California that hone in on only one of those conifers. So you have the F group over here, the P group only on the pines. Even though they're on the same slope in the Sierra Nevada, they're only affecting certain trees. And if you take an F1 and put it onto a pine, it won't work. If you take a P1 and put it onto a fir, it won't work. They don't do well. They don't interbreed. Okay? So it turns out the same three strains are in Norway or in Scandinavia, and the same story holds. But if you take an F from Norway and bring it over to a P and put them together, they will interbreed. A P from California. So there's something going on sympatrically where they're beginning to separate into new species, three different species, and probably based on their host. It hasn't happened long enough ago for there to be true separation. So if you take the one group from Norway and made it with a group from California that should be a different species, they're not. They're able to interbreed. So it's sort of we're catching it in action in that case with these microhabitat differences. But separated by enough space, they seem to be not separated into different species or strains, SP and F. Kind of complicated. The reason I don't bring in an example because there aren't really any good, clear images of this. But I was fascinated because I was all during the time on trying to figure out what the heck is a species, and then this study comes up, I'm like, great, okay, that clarifies that. I still don't know what's going on here, which is good. Gives me job security and something to do. Okay, now we have something going back to this adaptive radiation and getting into an island. Here are our Galapagos finches again. And the cartoon on the left here shows you all these different species. And you can get a sense that they have sort of honed in on different ways of getting resources. The ground finches, the ones up in the trees. Here's our woodpecker finch. Let's see, let me make sure you see this laser pointer going across to here. And you can see it's using a twig and poking into an ant hole or some kind of insect hole on this thing, much like a woodpecker would, but it doesn't have that long bill. And then you've got, yeah, these are seed eaters with huge beaks. This one down here at the lower right portion, etc. Here's the woodpecker finch in live action. You can't see the twig, but it's there over on the right. And then some large-billed, seed eaters. And so in theory, right, that first, or maybe it's had a few new ground finches migrate over from South America, and they start to interbreed and then slowly become separate breeding populations and then separate species. And they start to flex their muscles so much in a crowded space that they hone in on different resources and then really start to show differences in behavior and morphology and become new species as we see it today. This has happened in a lot of areas. I mentioned the Anole lizards in the south of us, that little sea called the Caribbean. There we go, not the Mediterranean, which was floating through my head. Here are the Silver Sword Alliance in Hawaii. So they are composites, sunflower group. I'll show you an example of the flower in a little bit, and you'll see, yeah, that's a sunflower, I see it. They can be arboreal, looking like a tree. They can be shrubby. They can be sort of pin-cushiony up on the top of the volcanoes. That's the form you see. Tends to survive better under the conditions up there at the top of the volcano. They can be what I call Dr. Seussy, a little stem with a flower raceme or something at the very top, a tuft of leaves down below. Sorry about that. And they can be just little things that grow in crags, like over here. All of these are very closely related to each other, and you'll see evidence of that in a minute. And they all evolved out of one little plant from the west coast of the United States, whose seed made it over to Hawaii, again, maybe more than once. The plant is called Media sativa, and it has these little tack-shaped glands that grow on it. Tar weed is a name that we use. I used to walk through the chaparral to my research site, and my boots and pants would stink like tar that night because I'd been walking through tar weed. Those little tack glands, or glands that are exuding this little tarry substance, really smell like tar. It's the tar weed. One of these, or maybe a couple, found their way over to Hawaii and then diverged according to different islands and different niche space over there. And that's how we get all those different forms. I'm going to go back to the previous slide real quick, I think. Yeah, and these are called Dabadia. It's a genus Dabadia, all four. Looking quite different, basically, from the pincushion from the arboreal type. Then we get the Wilkesias that also look quite different. So those are different genera by somebody's, the people that study them, definitions, okay? And you can see here's the Wilkesia, and these are the Dabadias, but there's also a group of Dabadias down here, and then there's these interlopers over here, starts with an A, and I don't know what their genus name is. But here's the Media down in here. So it looks like everything evolved out of the Media group. All right, and this is based on chloroplast DNA, which is sort of the go-to region for plant evolution. This is Bruce Baldwin, not the plant, but the guy. He did a lot of this work. Agroxiphium, that's what the A was. Sandwichiensis, subspecies Sandwichiensis, that's what this one up here is. They're beautiful, and they're a huge attraction, tourist attraction up on the craters of the Big Island and also Maui, Haleakala crater. And these look like they could really hurt if you fell on them. And when I would run through the chaparral as a kid in Southern California, yeah, we'd come out with our jeans totally ripped up from true silver swords that have sort of serrated, hard leaves. These things break, they're succulent, they're super soft, like there's that herb that grows in gardens that has that really soft fur, white fur on it that is called cat ear, I think, or something. These things are super gentle, definitely not a problem, versus the one in California is called the dagger, something dagger, and it is, you could really get hurt. Anyway, note that this here is a bunch of flowers, and each one is a little sunflower. Looks like this. It's got ray flowers and disc flowers, just like a sunflower does that you grow in your yard. Obviously a different variation on the theme, but it's a composite of lots of little flowers in one head. Now, they have found out that they can cross these things. So you could take a Dabadia and cross it with a Quihostia that's already been crossed with a Carlquistia, right? And you get a trigeneric hybrid. So what's a species in the silver sword group? They've only recently radiated in evolutionary time. Therefore, they haven't developed really strong separation of species and separation of the ability to interbreed. So there's a lot of interplay going on, but mostly they've honed in on different ecological habitats. So ecological species concept works, biological species concept, not so much, right? Okay, and yeah, orchids are like notorious. You could crossbreed genera of orchids like it's just not even thinking about it, if you will. Also, a lot of rapid radiation in the orchid group. And then we have, this doesn't happen just in, well, the hybrid issue doesn't just happen in plants. Here's a good example of a hybrid that's developing or is produced when you mate a blue spotted with a Jefferson salamander. And in this case, you get polyploidy, and plants are doing this left and right. And there's a researcher or a lab over at SU who, that's their bread and butter. They're looking at polyploidy of plants, and PhD student has done some really cool work showing how that impacts a lot of things with that plant. The polyploids actually are able to continue reproducing amongst themselves, but they can't back cross to the normal genetic background, which would be not polyploid, but the other version. And so that Tremblay's salamander is a hybrid, is a polyploid, can mate with itself, but it can't mate back to the blue spotted or the Jefferson. So that's a new species in one generation. You produce a bunch of Tremblays, they start mating, and you're good to go. So a lot of people think evolution is very gradual, and it is. And the gradual version is hard to catch. It's hard to actually get good solid evidence that a new species has developed because it takes so long, right? The ability to do a study to see that happen is sort of limited by our ability to live across that whole period of time. Geologic time, evolutionary time is much longer than the human lifespan or a human grant that allows him to do research, or her. However, these hybrid events, as I said, one shot, boom, you've got it. And that's a new species. So yeah, species are out there. All right, so that's a quick overview of the macroevolution. I hope I convinced you that it is a thing in today's parlance, that we do see new species, and we can basically rely on the fact that all those things out there have evolved since the beginning of the planet. Lamarck said, and others will say, no, they've been created as they are and have been here all along. And a Darwinian evolutionarist, or others, neo-Darwinian and other variations, will say, no, new species are constantly developing over evolutionary time. It's not static, right? We have allopatry and sympatry. You can have new species in a single generation through polyploidy. And with that very cursory overview with speciation, I'm going to move into microevolution in the next segment, where we'll look at, within a species, shifting phenotypes based on the environmental pressure, selection pressure of the environment, natural selection. Not creating a new species, but creating new variations of the species we see easily documented. All right, so very good. We'll see you at the next segment, and have a good next phase of your day. Cheers.